Okay, thanks very much to FMS for inviting me to talk about this today. I'm sure if you haven't heard about pink salmon and then the invasion of pink salmon into UK freshwaters, you must have been living under a rock somewhere, I think. It's a big topic issue. You'll see here it's not just me that's talking, it's Colin Bean et al. There are a group of people involved in this. The Pink Salmon Task Force has been sort of mentioned before. It is a group of dedicated and sad individuals, including myself, I would say. There's a picture of the individuals involved down here. There is one person who managed to evade getting their picture on the internet, and I commend them to MI5 if they're watching this. But it is a partnership. It's a partnership of government agencies, and fishery stakeholders. So that's largely led by FMS, but it does include representation from some individual district salmon fishery boards, soon to be joined by the Atlantic Salmon Trust as well. But that's how it actually stands at the moment. So, pink salmon. Obviously, you're probably well aware that the current issue we have with pink salmon has its origins in the attempts to establish a wild pink salmon fishery in the White Sea area of Russia. This started way back in 1956, and we tend to focus very much on the Russian introductions. But there was also other introductions of pink salmon around the globe around the same sort of time, largely in America. So there was one in Newfoundland in 1959, and at the same time there was a similar introduction into the North American Great Lakes. And the North American Great Lakes one is quite interesting because it actually demonstrates the plasticity of pink salmon. So this is a species which is anadromous, in the same way as Atlantic salmon is. But in the Great Lakes, it spends its entire life cycle within a freshwater environment. So it is a very adaptable animal. We have these odd year and even year populations, each with different sort of ecological tolerances. So they are quite different in many ways, or species that we really need to keep an eye on. But it was really in 2017 that things started to take a bit of a turn. So we've recorded them in Scotland since 1960. And these were generally animals which have been collected in sort of odd years, low density, odd sort of almost curiosities, if you like. And in the 56 years between 1960 and 2016, there were only 17 pink salmon ever recorded in Scotland. Some of them came from coastal fisheries and some of them were caught in rivers. But it was in 2017 that things really started to take off. We've seen this massive explosion of pink salmon around Northern Europe, particularly in places like in Norway and Finland in particular, but also here. So we went immediately from this 17 fish over that 56-year period to 171 in a single year. And so we would expect that number to increase if this was an invasion, every second year, every odd year, if you like. And these data sort of, to some extent, reflect that. So we had a big year in 2017, nothing in 2018, as we might expect, because this is an odd year, a population that was established. 2019, relatively fewer fish, but that was a very wet year. Doesn't mean to say that fish weren't there. It was a very wet year, so there might have been fewer people on the river. They might have been more difficult to record or to see or whatever. 2020, no fish. Then 2021, we had a bigger increase than we had in 2017. And in fact, I would argue there that, all of these numbers are very much underestimates. These are just numbers which have been recorded on the FMS app, and I'll talk a little bit about that in a second. And then we came back to another wet year in 2023, and of course that was a fewer records again, and most of them were actually made up from a couple of observations in one river, which fish weren't actually caught, they were seen. We see a sort of the patterns almost reflected in England, and you'll see the numbers in parentheses actually refer to numbers of fish which were caught in coastal nets. These were fish which were caught in the northeastern Northumbrian nets, and you can see there's some really significant numbers there. But in terms of what this means in terms of individual rivers and distribution, over the years. Well, I think that in those 56 years from 1960, only 15 catchments were affected there, with only 30 fish across the whole of the UK, 2 of those fish from marine environments. So fast forward to 2017 to 2023, a significant increase in the numbers of catchments which are affected. I mean, that's gone up by a factor of 300%, but the numbers of fish have actually increased by kind of almost 1,400%. So we've got just short of 400 fish, and 218 of those come from the coastal nets. So we have the app. So this was developed by FMS in 2017. It was the first of its kind, and that's fantastic because it gets us information on location, the data capture, the time of capture, the method of capture, whether it was just seen visually or actually captured. And there's also a kind of an option to provide a photograph of it for verification purposes. And that's great. So that gives us some really good basic information. It tells us a little bit about the numbers of fish. We can get information on the size, the weight, the sex ratio, and also some indication of the spawning condition of those animals, whether they're spent or not. And some of those visual records refer to the location and number of reds, and also the presence of dead fish as well, because a lot of those records aren't of live fish at all. It's actually records of dead individuals. So that's really useful. It's good information, and it's allowed us to kind of maybe sort of put a little bit of bones on what we expect an annual life cycle for pink salmon to look like. So we know there that we have fish arriving in the coastal zone when they're very silvery and very grouse-like, will we say, and there's an issue there about correct identification of these animals. And then when they enter the river as well towards the end of June, slightly after the arrival in coastal waters, we know that they're coming up to spawn, and they change their shape. We have the male with the characteristic hump. Both of them are kind of olive green with a kind of a pinkish tinge on them. And then those fish will spawn largely mid-August, early September. We'll see lots of these mortalities, so they should be coming up in the app. And then, of course, we know we have successful spawning in Scottish rivers. And we also know from the work done by Michael Scora and the Pinkses project that there was fish, or smolts, these pink salmon smolts, which are only 32 millimetres in length, really, really small fish, appearing in Scottish rivers around kind of the early part of the year into March. So we can even with the information we got from the FMS app, we're able to kind of try and piece together, if you like, a kind of a life cycle. So the app, it gives us information on how fish were captured or what state the fish was in when it was seen. And this, to the right-hand side, is actually a record from the whole of the UK looking at the prevalence of pink salmon around the coast using all records. So unsurprisingly, we have these records in the coastal area from the Northumbrian nets. But one thing I want you to take away from this is that most of these records are actually in the East Coast. Does that mean that pink salmon are targeting these systems? Is it simply because these are the first rivers they come across? Is it something to do with these are big, large rivers to do with discharge? Are they attracted to them? Is there something to do with catchment chemistry? We don't really know. It could even just be by chance. Okay. So one thing I would say there is that these are mostly records from the app, and I would sort of ask whether that reflects a degree of bias. I would suggest it probably does. So we need to sort of try and rectify that if we want to have a kind of an effective monitoring strategy. So we need to have something which is less biased towards where anglers are, I think. So we've got the app, which is great. It's very cheap. Doesn't cost them to run. It's already established. People know about it. And it's useful, but it must be spatially biased. So we thought, well, how can we actually rectify that? And we thought, well, actually, there actually are ways that we can use more modern techniques to try and expand that and make the programme a bit less biased. Certainly not going into the degree of statistical rigour that Ian went into earlier on this morning in terms of NEPs, but certainly just in terms of trying to get this more spatially sensible sampling programme. So eDNA can be scalable. We can do as little or as much of it as we possibly can. We know it works, it's field tested. We can use it to validate some of the records that are being provided on the app. And as I say, we can be reducing a spatial bias. And actually developing that programme was quite difficult in terms of how might we actually select rivers to do this? And we looked at various ways of doing that, and that eventually came back down to effectively fishery districts to make sure that we had at least one or more sites in every fishery district in Scotland. So you'll all be aware of how eDNA works. It's no big secret. You probably sat in a multitude of these conferences and seen slides which are broadly similar to this. But what I want you to get out of this is that in actual fact, because this is a partnership programme, there are some elements there where FMS took a very active role along with colleagues in SEPA. And that was largely around eDNA sampling and to some extent, some of them may have done some filtering, but that's where the kind of legwork is done, actually getting around these places and actually taking samples. We don't just go and take, a sample, a single sample of water. It's a bit more involved than that, as people who have done this will attest. You have to take multiple samples, you have to take controls as well. It's not an immediately simple process. So after filtering the water, and that can be difficult in particularly turbid environments, extracted DNA, the DNA is amplified, it's sequenced, it goes to the barcode library, and that tells you whether pink salmon are actually present or whether they're absent. Okay, simple as that. There is no big science involved in that. But so what's the best time to do this? So when should we do this? Well, so pink salmon will be sloughing eDNA or DNA from their bodies from the outset. They'll be doing this through their entire life. So they'll pick them up in coastal waters. You pick it up in fresh water as adults as they're moving in. Spawning's a particularly great time because you can imagine they're producing eggs, they're releasing milk. This is prime time for eDNA sampling. And of course, when the fish die and they decompose, of course, lots of eDNA from that as well. But we also have to remember that eDNA can be moved. It can be moved by things which eat fish. So scavengers and predators can also move eDNA around catchments, okay? So occasionally you might find eDNA in a place where you might not expect to find it. And there is quite, there are causal reasons for that. And of course, because we have eggs and in the gravel over the late part of the year, those eggs are, they are excreting metabolites and all the rest of it. There's eDNA alongside that. As the eggs hatch, they're releasing eDNA there as well. And of course, the same applies to alevins, and it also applies to the smokes as well. But timing is absolutely crucial because eDNA doesn't last in the environment forever, right? It can last for long times, and if we look at ancient DNA in the sediments of water, but we're talking about rivers here. So the key things that we have to think about here are the breakdown of eDNA with UV light, with high temperature, low pH, high flow. So if the water's, there's lots of water and it's running very quickly, it can take the eDNA with it. High turbidity as well, that can really have an impact on the filtering of water. And also the fact that if you've got low numbers of fish, it can be very difficult to actually pick up that signal. So how did we actually sort of move forward with this? So we kind of thought, well, okay, we've identified the rivers, so where will we take the samples from? We split that into two tiers. Tier 1 rivers were rivers which are sites where samples were taken 1 kilometre above the tidal limit and then 10% into the bottom half of the river length. And those samples were taken by SEPA staff. And then the remainder were taken by FMS biologists and District and Fishery Board biologists. And those were higher up the catchment. So these are our Tier 2 systems. So these are samples from the lower, middle and upper part of the catchment, but also samples taken from the confluence of major tributaries with the main river. And as I said, we've looked at a number of sites around the country. We had 31 sites in these Tier 1 sites, so these were the bottom parts of the rivers taken by SEPA. And we also had these Tier 2 sites, so there were 16 of those, and these are sort of a little bit more involved, so that's taking these samples in the catchment. We plan to take samples twice a year within the window of mid-June to mid to late August. One to try and determine the timing of entry, and another one to try and detect spawning periods. And the sampling protocol, we took at least 3 samples in every sampling sites, we also included a blank in that as well. And we used a couple of things. We could filter it with a syringe and a filter, or you could use a vampire pump. And of course, there's different efficiencies with that as well. So what did we end up with? This looks like a very messy map, and it'll get even messier in the next slide. But these are the 31 sites that we looked at initially. So the dots which are black are sites which appeared in the app. Okay, the sites in white, these are sites which weren't recorded in the app, and the ones in grey weren't recorded in the app, but there was some suspicion that there was pink salmon there based on other conversations with district salmon fishery board biologists and fishery trusts. So what's the kind of take home from this particular map? Well, one of them is there. eDNA we successfully looked at eDNA from pink salmon in the River Aire, where there was no record on the FMS app. We were able to confirm the presence of eDNA in sites where it did appear on the map, and I think that there was a couple of sites there where there was some suggestion that pink salmon were there, so they appeared in one or more of those individual replicates. And we extended that, of course, to the tier 2 sites. There was a lot fewer of those. But taken from this, we looked at for the Spey, for example, we had a positive hit for pink salmon eDNA in the lower part of that catchment, despite the fact that it wasn't on the FMS app either, and it was a suggestion or a suspicion that pink salmon might have been there. Put all this together, it comes to a very complicated sort of way of actually trying to pull this together. But I think suffice to say, that it's starting to show there that pink salmon has or eDNA has real promise in detecting pink salmon and using it to verify records where they exist, and also to identify rivers which didn't appear to have a record, where pink salmon were probably present. That's probably better. Those maps are sort of probably better expressed in this table. And so you see there that, as I say, just to kind of regurgitate this. So the Thurso, for example, it was on the FMS map. It was positive with eDNA. The Tweed is quite an interesting one, actually, because although it has the highest number of records on the FMS app, it didn't appear in the tier 1 sites, but it did appear in the tier 2 sites. There is some doubt about the validity of that record, I have to say, and I'll come back to that in a second. But I think hopefully you'll get some sort of indication that we are getting somewhere with this, and this is a kind of a learning by doing exercise. So no plan ever survives contact with the enemy. I mean, I think we're all probably aware of that particular phrase, and this is no different. So the Pink Salmon app yields important information. I think there is absolutely no doubt. That's fantastic. So we get lots of really good information from that. But the problem is that not all fish are entered into it. So there are some records or some observations that should have been in the app, weren't on the app. And of course, you can't enter something in the app if you haven't actually seen the animal. So there's some difficulties with that as well. But it could be spatially biased. I think that's fair enough. The molecular approach is effective. I think we can all agree on that. Particularly if we're sampling over large geographical areas. But we need to have this proper design. It has to be designed properly to give us the outputs we actually want. So we had to eliminate that spatial bias. And I think we would have. It's told us a lot about kind of how pink salmon are actually distributed in Scotland versus what we actually see in the app. And so the collection of some of these eDNA samples, we think probably could be integrated into some sampling programs that are already operated. So we know that SEPA, for example, go around sampling water around Scotland on a regular basis, and I wonder if we can actually try and try and integrate some of this sampling into those programs. So there may be some savings to be made there as well. So training fishery biologists was pretty simple. You know, it's not really that difficult. We're not taking them into the lab and showing them how to amplify DNA. It's about collecting the samples, but that has its own protocol to avoid, and contamination is a big issue and all the rest of it. So that's not a difficult thing, and that was successfully completed. But probably the biggest problem here, I think, is about providing time to purchase equipment and make sure we have funding in place. So FMS managed to get some money from the Marine Fund Scotland, which was really good. It was fantastic and all the rest of it. But we need to have that money in place in time to buy that equipment, because there's a lead-in time to actually buy stuff. You know, particularly when you're talking about lab consumables and things like that, and that caused a bit of a problem. So that was one of the biggest lessons, I think, you know, as a salmon task force, I think we learned in that process. The other thing is, you know, we're not the only country that are dealing with this, you know, and everyone is collecting pink salmon data using a completely diverse and really interesting way of doing it, you know, and that's everything from, you know, kind of certainly with the Norwegians collecting data at sea, ROVs, nets, direct observation, catching in nets, fish counters, angling records, visual records, use of ROVs or you know, kind of drones and things like that as well. And certainly Scotland's not too far behind that. We've used there's a variety of methodologies used to add information to the app. Certainly further ahead than many of our other colleagues around other parts of Europe. Certainly the main players here, I think, in terms of developing methodologies have been the Finns and the Norwegians and ourselves, I have to say. But so they're all experiencing, you know, kind of these invasions to greater or lesser degrees, particularly Finland and Norway, I have to say. And they use these different approaches. But there's no coordinated approach to monitoring, and there's no standard operating procedures in place. They're largely absent, so that makes, you know, kind of comparing data quite difficult. And of course, we've already said there's reasonable agreement with the eDNA-based approaches, but not everybody does it. You know, so just even looking at this, so we have the Swedes are involved in eDNA work, the Finns, the Norwegians, ourselves, and the Irish. Other parts of the UK, so Northern Ireland, Wales, England, don't intend going down that route at this moment in time. You can understand that from some of their perspectives, given the very low numbers of fish that they're actually experiencing. But anyway, certainly this proof of concept of using it as a monitoring protocol has been used by other people as well, so there's lots to learn there. So just on to my last slide with this. I mean, I think that, you know, where should we go? I mean, I think, you know, kind of optimising the Pink Salmon app is something we need to really go and make sure that we actually target, you know, good quality data and encourage better uptake of that to make sure that people actually do use it. The other things about funding, I mentioned the Marine Scotland, the Marine Fund for Scotland funding, which is fantastic. But I think one of the issues that we maybe come across is that, you know, kind of it's very difficult to get money for a pressure which is relatively new and maybe not causing that big of a problem right now, but it could cause a problem in future. So kind of where's the case for funding for these things? So actually, funding's quite crucial here. Need to have it in good time. Certainly, you know, kind of we're learning a lot from our colleagues in Norway and Finland, particularly from Norway, who spent a lot of money on this. So we kind of want to learn from others, and of course that comes down to developing this better cooperation and partnership and data sharing. So NASCO have just formed a new Pink Salmon Working Group, of which we're a part. And this will be launched at the next NASCO annual meeting in Ireland in June. But we're already, through the process of that group there, started to develop these this wider process of data sharing, and it looks good for the future. Not entirely sure whether it looks good for the future in terms of restricting the numbers of pink salmon coming to our shores, but certainly in terms of sharing data. And I'll finish there.